Well, good evening, ladies and gentlemen. Having shown you a few photographs of Steve Benner in his lab, it's now my pleasure to introduce the man in the flesh. So my name is Paul Davies. I'm director of the Beyond Center for Fundamental Concepts in Science. And as many of you know, our motto is confronting the big questions. And the sort of big questions we like to confront, I often say, are the things you love to talk about over dinner parties. Things like, how did the universe come to exist? How did life come to exist? Is time travel possible? What is the destiny of mankind? Where do the laws of physics come from? All that sort of stuff. And the question about how did life begin is, of course, one of the most fundamental that people can ask. For thousands of years, human beings have dreamed about being able to create life. The physicist Richard Feynman famously wrote, "What I cannot create, I do not understand." Creating life might be a dream, but people also fear its consequences, as Mary Shelley's famous story of Dr. Frankenstein's monster so graphically illustrated. Today, we no longer believe that life is some sort of magic matter, but rather an exquisitely fine-tuned and complex physical and chemical system that can be understood by the scientific method. But still, the prospect of designing and creating new forms of life remains both thrilling and, in some ways, troubling. And no person is better qualified to address this topic than tonight's speaker, who's come closest to attaining that goal. Stephen Benner established the Foundation for Applied Molecular Evolution in Gainesville, Florida, after a distinguished career in academia. He has a PhD from Harvard University in chemistry. I was tempted to say he's not merely a chemist, but some of my best friends are chemists. Steve is much more than a chemist, I think. His work has impacted fields with tongue-twisting names like paleogenetics, evolutionary bioinformatics, planetary biology and astrobiology, but especially the burgeoning new field of synthetic biology. His book, Life, the Universe and the Scientific Method, takes a penetrating look at the problem of what is life, and how would you know if you found it, for example, on Mars. Copies of the book will be for sale after the lecture, and Dr. Benner has kindly agreed to sign copies. One last announcement, please turn off your cell phones. And ladies and gentlemen, now join me in welcoming the 2004 Beyond Annual Lecturer, Dr. Stephen Benner.
Well, thank you very much, Paul. I'm afraid I'm surprised to see so many people here on a Friday evening. It speaks well, I guess, of the intellectual atmosphere in Tempe, Arizona, and surrounding communities. I'm hoping I'm not going to just disappoint. How are we doing? Can people hear me, or would you like it louder? All right. Excellent. So what is life? And the problem is my next slide is going to be perhaps, well, perhaps it won't work at all. Don't know. But it seems like this is the kind of question that should be approachable using what we teach in middle school as the scientific method, right? Because after all, if we're going to go search for life on Mars, should we not know what we're going to be searching for? And it seems to me, therefore, that the scientific method should be brought to bear to answer the question, what is life? Now, who here did a science fair project when they were in middle school? Raise their hand. Who enjoyed it? See, the problem is that, we're not going to get a lot of information for this kind of a big question from the science fair middle school paradigm, because, if you're in this game, what you are supposed to do is ask a question, then you do like background research, and then you construct hypotheses which you test with an experiment, a deftly designed experiment, so that even a negative result tells you something informative. You're supposed to analyze results, draw conclusions, and if the hypothesis is true or partly true, you're supposed to report the results, and if not, you're supposed to go back and think and try again. Now, That's fine, but it's a skill. There's no question that it's like playing chords on a piano, right? If you're going to try to learn how to play the piano, or if you're trying to learn how to do science, presumably you have to be able to do something like this. But it really doesn't do much for these big questions, like what is life, or how did life originate, or how might we recognize life if we were to encounter it on Mars. And so I thought I would sort of weave these two questions together tonight with sort of a semi-philosophical look at what we do as scientists to try to ask some of these big questions. And of course, the very first thing that we do is we observe. And this is certainly true for our friend the biologist who is observing a moose. That is a biological living entity, and there's no question that he is going to consider that moose the subject matter of his discipline. Certainly a moose is within biology. Of course, one of the problems already is that you have two kinds of biology. There's the so-called natural history biology, which means that you observe biology, you observe animals, you observe moose. Maybe you might look at the fossils, and then somehow maybe you might probe them a little bit. So a good friend of mine, Gary Bolofsky, would go out into the Montana plateau, pick up moose droppings, figure out what the moose had been eating, and then make an energy balance. Try to predict how the moose would be, walking around the next week or the next month in response, say, to environmental stress. Of course, the biologist has this choice. He can also do something different. He can shoot the moose, right? And this is why this is a Jake Fuller cartoon of decisions that biologists make, because if you kill the living things and you can analyze its pieces, you might be able to go further. Of course, the natural historian biologist doesn't think that this is biology any longer at all. Right? And of course, this is no love lost between these people. Harvard's biology department divided it into two because the two groups of people, those who observed the moose and the people who shot the moose, could not really get along. But the bottom line is that observation alone in biology has not turned out to be particularly productive in going past this sort of bulk descriptive. Describe the moose, you can describe what he eats, what he does, but it doesn't really answer the why of the moose. So there's analysis, okay, and so the analysis, of course, starts with, unfortunately, killing the organism. It's then dissected. The dissection is to the level of the organ, and then, of course, the cell, but eventually to the level of the molecule. We have, of course, the historical beginnings of analysis in modern biology, where they're trying to look at the mastodon and decide what to do with the pieces. But at some point, you get all the way down to molecular representations of what is in the moose or the mastodon. In fact, you're looking at this here. This is a structure of something that you have in your bodies. It's called the ribosome. It is actually the molecular machine that makes proteins. You're making proteins all the time from the food that you are presently digested. Of course, you ate protein. The protein is being broken down into amino acids, and they're being reassembled into new proteins, your proteins, not the mastodon that you had for dinner. And so the bottom line here is that at some point, analysis gets to the point where you're actually looking at individual atoms, and that's what these little bumps are. Each one is a different atom, and that's dissection roughly to the level of the molecular system, which makes most biology today associated with a chemical description. Almost everybody will talk about metabolism. They'll talk about the chemistry of metabolism. We talk about vitamins. They'll talk about the molecules that are vitamins. They'll talk about genetics. They talk about the DNA that's part of genetics. And so that's a very popular area. Of course, it's also an explanation for why we do not find aliens among us, because as any alien will tell you, if you get a scientist near an alien, what does the scientist want to do to the alien? Well, they want to dissect him, and there's 12 Steven Spielberg movies embedded in that observation. Great. Now, keep in mind that the analysis plus observation together has been quite productive. We do have theories for life, and I've represented 3 of them here by the appropriate statues. There's a cell theory of life, roughly 1838. This is a guy named Schwann, who is a man who said, hey, maybe, actually, it's kind of amusing. If you read his original papers, he's trying to unify botany and zoology, noticing that both animals and plants have something in common, that is that they are made up of cells. And so this is the cell theory of life, and one of the leading journals in modern biological science is called Cell, simply Cell. Of course, this is Charles Darwin as a young man, much more handsome than those pictures that you see of him as an old man. And he has evolutionary theory, which is, of course, a very central theory in biology as well. Of course, eventually, here's Watson and Crick talking about the gene theory. There is their famous structure of DNA. I've expanded some of the DNA into chemical structures here. And so we do actually have from analysis, we have from observation these theories. They exist, and people talk about them, people teach them in classes. And so you can come up with sort of this idea that sort of combines them. This is actually a definition that was suggested by none other than Carl Sagan, which is being used, among other things, as NASA goes out and looks for life in the cosmos, that life is a self-sustaining chemical system capable of Darwinian evolution. And what we mean by self-sustaining is not that you can live without anybody else around you. You have to eat things, but you are able to go out and get your own food, right? And of course, capable of Darwinian evolution. Darwinian evolution is a big term with lots of baggage. It means being able to have children. The children are mutants sometimes in some detail. Actually, many people think they always are, but to some extent they are always. And then, of course, having a survival of the fittest and natural selection operate on it. That's all embedded in those two words. Well, excellent. So Watson-Crick model, I'm going to be this dangerously close to chemistry, but I'm going to try to help you out here. There you go. I mean, Watson-Crick model for DNA. Almost everybody saw this at some point in their, okay, so who saw this at some point in their high school education? Raise your hand. You see, now that's a good response. That shows the robustness of the American education system. Fair enough. A pairs with T, G pairs with C in this double helix, where replication, and there's the double helix over here. Replication involves separating the strands and then making a complementary strand by putting in G opposite C and T opposite A and A opposite T and so on. And you get two copies of the same thing. Occasionally there's a mistake. That's the mutation, which if it's not repaired ends up being the playground of evolution. But it's a relatively straightforward model, and it's actually 60 years old, roughly. So now you can ask the question, so how has observation and analysis and these theories of biology served us when we actually needed to do something with them, like look for life on an alien planet, in this particular case, Mars? Now, 1976, about 40 years ago, Viking mission, actually was two duplicate missions, landed at two separate sites on the surface of Mars. They were at the time roughly the most expensive experiments, scientific experiments that were ever done. And they carried with them life detection experiments. And in fact, there were 3 of them. And you can think about this for a minute, because the first one, they were going to sprinkle food on the surface of Mars and see if carbon dioxide is emitted. That's the purple line. Second, they were going to water the surface of Mars and see if oxygen was released. The third thing they were going to do is they were going to put carbon dioxide on the surface, in the atmosphere above the surface of Mars, a little bit of carbon monoxide trying to reproduce the actual atmosphere of Mars. They were going to see if carbon entered the ground, was fixed, as we like to say. Now, these experiments actually are not applying any of those 3 theories of life that I've told you about. They don't apply a cell theory. They don't apply an evolutionary theory. They don't even apply a gene theory of life. What they are doing actually is constructively applying what we would call a metabolism theory of life. And the reason why is because if I go out and feed you some food, you will release carbon dioxide. If I water the ground outside where something is growing, photosynthesis will take place and it will release oxygen. And of course, terran organisms at the bottom of the food chain will actually fix carbon dioxide. That's how the plants get their carbon, right? They have carbon dioxide in the atmosphere, which they suck up and make into plants. Now, you can look at that and you can say, this is what we call terracentric or earthocentric, right? We are making the design, the test, the looking for life on Mars. We're designing it, expecting the life on Mars to be very much like the life on Earth. And for those of you who are not aware of these results, all 3 tests for life on Mars were positive in 1976. This is Gil Levin. He's the man who designed the Rabel release experiment. He will still tell you, he's still living, he will still tell you that he is the man who discovered life on Mars. The conclusion, therefore, all CO2 was released when they sprinkled food on, the oxygen was released when they watered the surface of Mars, and carbon was fixed when they put radioactive carbon on the surface. The conclusion, life is present. That is, if you're going to interpret these tests on their own terms, there's life on Mars. And so the question you probably are wondering is, why the hell haven't I been told about this, right? Who thinks that there is life on Mars? Raise your hand. Who's Gil? Is Gil in the audience? I don't see him. Right. What's that? Yes, absolutely. He has got great associations. So the bottom line is that those experiments were tested. So the question is, why haven't you heard about it? Why? I get many more hands up when I say, who's seen the structure, double helix, than I get by having somebody who's heard that these 3 tests were positive on Mars. And the reason for this is because they did another experiment, all right? It took a little while. This is a gas chromatography mass spectrometer. For those of you who are in the business, you know exactly what this guy does. They scooped up a little bit of Martian soil, they emptied it into a cup, they heated it up to various degrees of temperature, and what they're looking for is gas coming out, which they flow into a hydrogen tube, and then they have a palladium separator and all the rest of it. But what they're looking for is reduced organic molecules, well, like glucose, or its products. If I heat glucose up to 400 degrees, I get something off, or protein. If I heat protein up to 400 degrees, I'll get organic compounds off. What did they find? Well, they found nothing. They found no organics. All right? So we have 3 positive tests for metabolism, and now we've got 1 test on the other side that said that there's no organic molecules. Well, no one said anything about an organic molecule theory of life. We talked about the evolutionary theory of life, the cell theory of life, the genetic theory of life, but now we know that they're operationally, constructively using the organic or reduced carbon theory of life, and we know it trumps, actually, a metabolism theory of life, because if you got 3 positive tests for metabolism and 1 saying there's no reduced organic, the no reduced organic wins. So you're learning something about how these scientists are thinking. But here's even worse. The minute they decided that there was no life on Mars, there could be no life on Mars because there was no reduced organic compound, what did they do? Well, the minute that they decided that there was no life on Mars by the GC mass spec, they went back and found explanations for how you could get 3 positive metabolic results out of the metabolism test. And of course, the first thing they noticed was this sort of Venn diagram, which is the red circle is all systems that transform organic compounds, and the blue circle is all living systems, including you. But of course, outside the living system circle, but still within the transforming organic species circle, is your car, and no one thinks your car is alive. So that was saying that, okay, as soon as the community decided that they needed explanations to find non-life explanations for the signs of Martian life, they found them, and they found them within minutes, days, not weeks, months, or years after the results came in. Now, you could have asked how come they didn't think of these beforehand, right? But that's a separate question, which I will entertain in the question and answer period. Well, this is water, by the way, just for those of you who want to know what they think happened, or what they thought happened. This has continued to evolve. Water is H2O. Many people know that. Ultraviolet light comes to the surface of Mars much more intensely than on Earth because it does not have an ozone shield like the Earth has. It will split water into an HO and an HO and an H dot and an H dot. These are 2 hydrogen atoms, each with their electrons. And then what happens is that these 2 H dots can recombine to give hydrogen dihydrogen that may escape into space, leaving behind hydrogen peroxide, which is a reactive species, is an oxidizing species. It's something you might have in your bathroom or in your mouthwash where you swish your mouth with it, you get off bubbles of oxygen. And of course, this is a reasonably powerful oxidant, or it can be. And so the argument was that this is what is actually oxidizing the food to make carbon dioxide, not metabolism. This is an inorganic oxidation process, getting its energy ultimately from the ultraviolet light that is coming from the sun to Mars. So there goes poor Gil Levin's test. There is the food that they sprinkled. They put a couple of amino acids like glycine, a sugar like glycolic acid, or lactic acid is not quite sugars. But in point of fact, all of these are oxidized by hydrogen peroxide in the presence of iron. By the way, this is a reaction that was known in 1900, discovered by the German chemist Ruf. It is called the Ruf-Abbau, or the Ruf degradation. It's a well-known species. Iron is the catalyst. Mars is the red planet, and it's red presumably because of iron in the surface. So the science is, and by the way, just for the record, adding water is one of the ways that was argued to activate the hydrogen peroxide that's left behind, and ultraviolet light was being used as an argument as how the carbon dioxide was fixed. So I don't need to go through the details. I certainly don't want you to walk out the door remembering the details. All that you should know is that the minute you had a theory, the reduced carbon theory, with a result, the gas chromatography mass spectrometer instrument, trump the metabolism theory folded, and immediately people found ways of actually explaining those results. And I don't dare go much farther than to say results mean what the culture needs them to mean. That's sort of how it happened. So the community was despondent. Okay, life was not likely to be present on Mars. Actually, this is, I should say that this book that Paul mentioned that's out in the lobby is we got the Gainesville Sun political cartoonist to do cartoons for us. And I must confess that all I said was that the community was despondent. Before I know it, Jake Fuller has generated this, where a nice Martian is holding something called the McKay Report. In 1996, the McKay Report came out to cheer up the despondent scientists who were unhappy that they were not going to go find life on Mars. And what was in the McKay Report? Well, we actually, by the way, in case you didn't know, have pieces of Mars on Earth. What happens is something slams into Mars, ejects rocks. Some of them are ejected with enough speed to reach escape velocity, and then they travel around the solar system for a little while, and some of them land in Antarctica, and then you go down and scoop them up off the ice, because anything that's on the top of ice is likely to have fallen in, and a small fraction of the meteorites that you find were explained as Martian-derived, and there are people look at atmospheric composition in those shot glasses and the Martian meteors and these type of things. This is Allen Hill, 84001. It's a meteorite from Mars. Dave McKay looked at it closely, and what he saw were cells. Now we're getting somewhere, right? We do at least have a cell theory of life, yes? And so now that trumps the organic theory, right? Something was wrong with the GC. Mass spec. Actually, it turned out that there was an entirely separate interpretation for why that instrument did not find. Now Gill's pretty happy, right? Because after all, now what you're seeing is a strong theory of life. You're actually observing cells. So the cell theory of life trumps the carbon theory of life, which of course trumps the metabolism theory of life. And so in 1996, Bill Clinton went on television. In fact, if you want to see the clip, you can actually see it in the movie Contact, where the Carl Sagan excerpted Bill Clinton's discussion of the discovery of maybe life on Mars into the discovery, of course, by Jodie Foster of extraterrestrial intelligence by their radio signals, for those of you who have seen the movie. So now what I'm doing here is we're being sort of anthropologists of science, right? So we're looking at the scientists and we're trying to make the distinction between what scientists say they believe and what they constructively believe. And remember, a constructive belief is different from an actual belief because you act on it. So who believes here that quantum mechanics is true? Raise their hand. Yeah. Now, for whom is that a constructive belief? Like, what did you do today that actually required that you believe that quantum mechanics was correct? Nothing. I mean, even Paul, you didn't do anything today that required quantum mechanics to be correct. Well, maybe you did. So you're looking at the scientists and seeing what they do. Now, for those of you who are students in the room especially, what did I forget to put on this picture? A scale bar, thank you. You're not a student. Absolutely. This is a microscopy 101. What we forgot to put on that picture is a scale bar. So I will do now here. That scale bar is roughly 1 micrometer, 10 to the minus 6 meters. There's a meter, a millionth of a meter. And so there's the size of an E. coli, a typical bacterial cell, given that scale bar. And people looked at that, and all of a sudden they said that these cells are too small to be life. Now, I don't know if that was a very persuasive argument to everybody who was a cell biologist, but the only correct response is what makes you think life cells on Mars have the same constraints that cells on Earth have, right? So you say too small? Too small? For what? Well, a presumed volume needed for a presumed set of components. Well, what could those possibly be? Well, there was an answer to that question when it was raised, and the answer was there's that ribosome again. That's that ribosome I showed you a few slides earlier, which is the machine that you have to make proteins. So there's the ribosome, the machine in Terran life that makes proteins. If I scale it down to here, it's roughly that big, which is roughly the width of that cell. And so the argument is that this cell is too small to be life. Now, we try to get the students to write out clean Aristotelian syllogisms. And so when you do so, notice that you miss a premise, right? So if you say the cell is too small for a ribosome, therefore the cell is too small for life, you're missing a premise, right? Because somewhere it's tucked in there, there's got to be a premise saying that proteins, which are what's made by the ribosome, are required for life, right? So maybe the cell is too small for ribosomes. Ribosomes are required to make proteins. Proteins are required for life. Therefore, the cell is too small for life. But you can't go, just hum the words to that. You got to actually say it all the way out. Because the question is, who is the third person who talked about the protein theory of life, right? We've talked about the cell theory of life. We talked about the genetic theory of life. We've talked about the evolutionary theory of life. We talked about the metabolism theory of life. We talked about the reduced carbon theory of life. We talked about the cell came back and trumped the metabolism. And now we're saying, life requires proteins. But there's a problem with that. And that problem is actually captured in the two colors of that ribosome. Okay, the two colors of that ribosome. I'm now spelling this out for you. We might do some logic. Proteins are made by a ribosome on Earth today. Today, the information, by the way, in DNA is used to make an RNA molecule carrying the information from the DNA. Then RNA directs the ribosome to make proteins. Of course, presumably life had to start sometime, and if you've got a system which requires DNA and a ribosome and an RNA molecule carrying information, that's a pretty complex system. And so it suffers from this question as to how did the system get started? Like, what are the odds that a DNA molecule emerged from Darwin's warm little pond? That's pretty low. What's the odds that a ribosome did it at the same time, as well as all the translation machinery? This is what my creationist friends call the irreducible complexity problem, right? You just can't get it started because there's just too much. And so, but never mind, the bottom line is it turns out that the ribosome, which we understand the structure by analysis, is in fact, it is the machine that makes proteins, but it turns out that it's not made of proteins mostly. The orangey colors are proteins, are actually RNA molecules, and the blue components are actually proteins slapped on the surface. So what this says, and by the way, where the protein is actually made is surrounded by RNA. It's the RNA part of the ribosome that makes the protein. So now all of a sudden you say, well, if RNA is needed to make proteins, then RNA came before proteins. Are you with me? Right? And by the way, some people have taken the leap that as an early episode of life on Earth used RNA as the only encoded catalyst. That RNA in the early life forms did both genetics and metabolism. One molecule doing both. That's actually a nice way of solving the chicken and egg problem, because if proteins are needed to make RNA and RNA is needed to make proteins, which comes first? Well, the answer is RNA comes first, and the ribosome contains the evidence of that, because in the ribosome is the machinery that makes proteins. Proteins before RNA. Now, if you think about that for 30 seconds in context of your discussion of the Martian meteorite, right? We said that the meteorite was too small to hold ribosomes. But if ribosomes are needed to make proteins, and it's the RNA of the ribosomes that made proteins, and the RNA therefore came before proteins, the assumption then is that there was not only is it possible for life to have no proteins, but that the earlier versions of life on Earth were organisms, RNA organisms, that did not use proteins at all. Now, you're going to have to walk with me through this. By the way, just for those of you who are into vitamins, a lot of the vitamins that you eat are in fact cofactors that have a piece of RNA, and the argument has been that it's left over from a time on Earth where all the catalyst, all the metabolism that you did was catalyzed by RNA species. So if you go out, for example, and buy yourself a vitamin with pantothenic acid in it, and many of you do, that's part of a cofactor that has an RNA piece. If you go in and buy something which is a part of coenzyme A, it's an RNA piece. ATP, the energy of life, has an RNA piece. The reaction can go just as well without the RNA piece. So the argument is that these are vestiges, as it were, molecularist pandas thumb, of a time where your ancestors, life on Earth, did everything that life needed to do without proteins, and this is built into the historical analysis. So now work with me here, okay? An ancestral form of life on Earth that used RNA as the only encoded biopolymer is, of course, a vestige of the thing that makes proteins. RNA before proteins means life without proteins, life with only RNA. So if the RNA part of the ribosome is the part that makes the proteins, therefore, ergo, RNA became before proteins. Double ergo, life is possible without proteins, with RNA alone. By the way, just for the record, about 70% of the volume of a big micron cell, the ones that cell biologists are used to looking at, the same cell biologists who think that the life that we see, the cells on Dave McKay's, Alan Hill's Martian meteorite are too small to hold life, about 70% of the non-solvent volume of a bacteria living in your stomach is being used to make proteins. So if you can get rid of the need to make proteins, triple ergo, oops, I went 2 slides, right? Life based on RNA alone can be much, much smaller, can be in much, much smaller cells than life that needs proteins. Quadruple ergo, therefore, the Allen Hill structures are not too small to be life. They just have to be life without having to devote 70% of their volume to making proteins, which is what the E. coli living in your stomach are doing. How are we doing? So there you are back. We put the scale bar in. There are the structures of Dave McKay. This cell, a modern bacterial typical cell, is large because it has to make proteins, therefore it has to have a lot of these. The argument, these cells are not too small to be life based on RNA only, a kind of life that we think was actually our own ancestor on Earth. And that's because they don't have to contain any of these guys, because they don't need proteins. So keep in mind, we're constraining our prejudice of what life is. And of course, just for the record, if you go out in there into the galaxy and you ask, if you actually do believe that there was a prebiotic world, that life at some point originates, this is like a hitchhiker guide, everybody agrees that this was a bad idea, but never mind. You make the RNA world, it then takes some time to invent the ribosome. Okay, the ribosome is eventually invented, and now you can start to have an RNA protein world. And of course, at some point, intelligence is invented, and therefore you can actually go back and use your RNA protein life to explore the cosmos. Well, what are you most likely to find? Are you most likely to find life that contains proteins like yourself? Or are you most likely to find the most primitive form of life, which only had one biopolymer in it, RNA? And the answer to that is it depends on how fast it is able to invent the ribosome. If it's slow to invent the ribosome is difficult to invent. If it's difficult to invent the machine that makes proteins, you go out to the cosmos, you're going to find life that uses only RNA as its genetics and metabolism molecule. Of course, if ribosomes are invented quickly, then of course, after the life is originated, then of course you'll find mostly organisms that use proteins. And of course, if you believe that intelligence is fast or slow, people have different views on that. But if you think that intelligence is slow to invent, you'll mostly encounter unintelligent organisms, whereas if intelligence is quickly invented, you'll find mostly intelligent organisms. So again, I happen to think it's a little bit of a trouble to invent this machine. Therefore, I would not be surprised if all of the life on Mars were not, in fact, RNA life. So are you with me? By analysis, right? By dissecting the moose, by dissecting the mastodon, we've come up with an RNA molecule, a ribosome, that makes proteins. From that, we say RNA, which is the orange part, which actually makes the proteins, is more important for making proteins than the protein part. RNA came first, proteins came later. Therefore, this kind of structure is early, proteins are late. Life is possible with this kind of structure only, and not with proteins being required. Now, sadly, the Martian cells turned out to be mineral ridges. You turn the rock on its side and you look at it a different way. And so the question of Martian life shifts back to the despondent side. But later, liquid water was found on Mars. And of course, that's the water theory of life, where if you have water, the water theory of life sort of trumps any of the things there. Clearly, and so on. Clearly the science is not supporting us as we would like, especially when we set out to seek weird life, right? Because we're constantly going back and forth and back and forth and back and forth. If the science were doing its job, you'd expect to converge on some sort of a solution. We haven't done that. All right, no problem. But there's a third thing that we can do besides observation and analysis. We can do something called synthesis. Now, what synthesis is a way of assembling things. You put things together in pieces. We've already done, as I've explained, the sort of exploration as we try to find examples of alien life that helps us understand what life is. We can already do an examination of fossils and modern life to try to understand what simpler life might be. This is indeed going backwards in time from the modern ribosomes, for example, to more ancient ribosomes. I won't talk today about any prebiotic chemistry where you try to get life out of prebiotic soup in the laboratory. But I'm going to talk now about the fourth approach, which is to actually try to construct life in the laboratory, right? There's sort of a reason for this, and Feynman was already mentioned this evening. Because if we truly understand what allows a system to sustain Darwinian evolution, we should be able to make one of our own, right? If you're so smart, right, do it, right, if you understand this. And this is, of course, the Feynman remark that was already quoted: what I cannot create, I do not understand. Another one of my favorite Feynman quotes is, people are easy to fool, and the easiest person to fool is yourself. Synthesis does a couple things for you. One of the things it does is not only demonstrates that you understand something, but it prevents you from fooling yourself into thinking you can do something, or from thinking you understand something when you don't. And that is a really important thing, because in my view, science is an intellectual activity that embodies a mechanism that prevents the scientists from always reaching the conclusions that they set out to reach. I mean, most of the time we're going to do that, and there's nothing we can do about that. But what science does is has an experimental method or an observational method, something that every now and then, once in a blue moon, gets the scientist to agree that he was wrong at some point, or she was wrong at some point. So synthesis is a mechanism for preventing you from fooling yourself. If your theory is inadequate, you cannot fool yourself into thinking the opposite. If you try, the synthesis fails, and it fails in a way that, that cannot be avoided. My favorite example of this, and I really should not bring this up too often, right? Keep in mind, is this Mars climate orbiter. Now keep in mind that the human instinct, and this is true even if you're a human scientist, what do you do if the observation that you have made contradicts your hypothesis? You don't discard the hypothesis, you discard the observation, right? And in some cases, by the way, that's entirely reasonable, right? Because if the spectrometer is broken and doesn't give you, right, the right answer because it's broken, you should discard that. Right? I mean, this is the classic example, is, you know, if all emeralds are green, so you just observe an emerald and you discover it's not green, does that violate the rule? Well, no, you observed it under ultraviolet light. Well, okay, I didn't mean that. All emeralds are green when you observe them under white light. So then you send out an emerald and somebody comes along and looks at it under white light, and he says it's not green. He has red-green color blindness. So you say, okay, I didn't mean that, right? All emeralds are green when observed under right. This is the called Quine-Duhem paradox. It's the fact that you are always making inferences based on a lot of assumptions that you don't really think about all the time. And so a lot of times, you know, you maybe should be discarding the observation rather than the hypothesis. But that's not a good idea to do as a general rule. And one of the classic examples is that the Mars climate orbiter, which is going out to Mars, where the guidance system hardware was operating under the English system, you know, feet, pounds, this type of thing. The guidance system software was being in the metric system. All the way out, if you read the mission logs, the people knew something was wrong. Right? They were burning too much fuel to correct it, and they were able to rationalize it away. And then, of course, but at some point, you're targeting this grand challenge. The synthesis is forcing these scientists across uncharted grounds. If you're wrong, the rocket crashes, and there's no way around that. And so synthesis is able to drive paradigm change in ways that analysis cannot, because as you are happily writing your grand applications, do easy hypotheses, ignoring data that contradicting them and publishing the data that confirm them, right? If you're trying to do synthesis, it puts a rapid end to it. So what we'd like to say is that synthesis sets this grand challenge. This is also in biology now. This is what synthetic biology is. You set this grand challenge, you drag scientists kicking and screaming. There Jake has the scientists being dragged across uncharted territory where they're forced to solve unscripted questions. Okay, unscripted problems using available theory. When the theory is inadequate, the synthesis fails, and it fails in a way that, that cannot be ignored. Therefore, synthesis drives discovery and paradigm changes in ways that observation and analysis not. And so now we're on to the next stage. Okay, so we think we're smart. We think that RNA alone should support a self-sustaining chemical system capable of Darwinian evolution. That's a definition of life, right? And so we ought to be able to do it if that's all there is to it. So there is a Watson-Crick base pair. Who here has ever taken a course in organic chemistry? Raise their hand. Who liked the course? I say, oh dear. Right. Watson-Crick base pairing is easy to understand, and I'll get it easier on the next slide for those of you who don't know chemistry. But basically, there's a big piece, 2 rings, and a small piece, 1 ring. Big pairs with small. That's one of the rules of complementarity. But then hydrogen bond donors, which I have in red, and pair with hydrogen bond acceptors, which I have in blue. So cytosine, the C in DNA, has a small thing with a donor acceptor red, blue hydrogen bonding pattern. And it pairs with guanine, which is a big thing with a blue, red hydrogen bonding pattern, acceptor, donor. And for those of you who are not chemists, let me see if I can go to this. We can go to that structure, which maybe puts that a little bit more clearly, right? It's putting together Lego sets, right? We have a big Lego piece and a small Lego piece. The big Lego piece has prongs and holes, and C pairs with G because it's got a prong, hole, complementary to the hole, prong, right? And contrast T. Well, God didn't get it right. I mean, God, even with adenine, forgot to put the prong on the lower Lego piece. So you actually hold the AT base pair together just with the top two hydrogen bonds. But that's a mistake that I assure you we do not make in the laboratory as chemists. We will put that NH2 group back. But never mind. Now, one of the things that you've got is a problem here because if you think about this as for 30 seconds, we like the idea, if we want to build our own Darwinian system from scratch, to demonstrate understanding, of course, not because we want to take over the world like Mary Shelley, whose protagonist, does. We like the idea that nucleic acids alone might be sufficient for Darwin, right? We liked, because that means we just have to design the nucleic acids. We don't have to also design proteins. So it's half the work. That way we only need to design one biopolymer, not two, from scratch. But there's a problem. People have tried this. I mean, people have tried to take, this relatively simple system, even missing, well, I put already the amino group back in for the chemists who are in the audience. People have actually tried to get good function out of this system. They've largely failed. We say that RNA should be able to support catalysis and genetics, that is metabolism and inheritance, without proteins. But the RNA just has 4 building blocks, DNA the same. So it's actually not so many. I mean, proteins have 20 different building blocks, and they have all kinds of 20 kinds of stuff on them. And the more stuff, the more you can do with a protein. So it's not terribly surprising that proteins are much better at doing metabolism. They're much better at doing structure in some sense. And so RNA has much less to work with, and so this is one of the arguments why people have failed to actually get. I mean, you can say if I think that life originated by an RNA molecule crawling out of a test tube or crawling out of a prebiotic soup and asking me to dance, right? I ought to be able to do it in the laboratory, right? But people have tried and people failed, and it's not clear why. But one possibility of why is you only have 4 building blocks and not much stuff to work with, whereas proteins have got a lot of interesting chemical stuff. So the goal now is to take this structure. We've got a hypothesis now, so I am back in secondary school, middle school science fairs, right? We have a hypothesis. If we add more building blocks to RNA and put more stuff on them, that the RNA will be better able to support Darwinian evolution. So the question is, are there any suggestions as to how to add more letters into the nucleic acid alphabet? Now, the people in the front row don't want to answer that question, and I'm not going to get anybody to answer this question until I start putting this back. There are the 2 rules in cartoon form. Big pairs with small. God's mistake, left out a prong on this one. We'll put it back in. But you'll notice that I have a red dot, a prong, and a red dot. I have a prong, a red dot, and a red dot. And I have not even begun to consume all possible designs of all Lego blocks, right? It doesn't take much to say, okay, there's T, small, dot, prong, dot, C, prong, dot, dot, complementary to the big thing, right? Dot, prong, and prong, dot, well, there's God's mistake. But I can also have a dot, dot, prong, which is complementary to a prong, dot, dot. I can make another piece of Lego brick, and these will fit together. In fact, this guy will fit together with this guy will fit together with this guy will fit together with this guy. It's sort of like looking at Braille, right? Figuring out all the ways you can have dots and prongs on these things. And so, it's great. We can also make up new letters, JV, KX, ZP, and my favorite SB. The bottom line is that you can do all these structures in principle. The chemistry is now that, right? There you are again now with the chemistry. With instead of prongs and dots, we have, of course, actual atoms and hydrogen bonding groups. And so, yeah, if it's so simple, in fact, that's what the Watson-Crick theory is. It's a very simple model for how genetics works. We ought to be able to get DNA not with 4 letters in it, but 12. Now, that's not quite 20, which is what proteins have, but it's more, okay? And we could, with more stuff, we can do more useful things. So what we're going to now do is do a new kind of genetic system that is not found anywhere on the on Earth, not that I know of. Although Paul might have some disagreement with that. I mean, it's a fact, by the way, and Paul has written extensively on this, that there could very well be the life I'm about to discover or to describe under the carpet here, and not because we made it and put it there, right? But it could be there naturally, and all the tests that we would use to look for it would not have found it. This is Carol Cleland's shadow biosphere, right, which should get you the willies if you haven't gotten them already. A new kind of genetic system, and just for the record, I won't go through this details. I certainly will not bore you. All of those molecules have been made. Actually, Stefan Lutz here is from Emory College. He sees Emory University. He sees one of the compounds that he worked on, which is that one, I guess. Made that tritiated, if I recall, for as part of his DHT. He gave the chemistry seminar at 3 o'clock this afternoon. So there you go. We made these molecules. We put them in double strands. Zhen Wang, who provided from Georgia State, provided us with selenium nucleosides to help us solve the crystal structure. And it looks just like the big, small hydrogen bond donor, hydrogen bond acceptor, exactly the things that you see. This happens to be the ZP pair. Chris Switzer has looked at the SB pair, and I won't go through all the details. But it really behaves like DNA. That's actually useful. It turns out that if I have AT and GC base pairs and I put them into your body, you got lots of DNA and lots of RNA with AT or U, G, and C. And so almost all of the nucleic acids in your body will interfere with DNA that you put in, right? And so we have a very common thing which we want to do is be able to detect nucleic acids in your body. Why? Well, because unwanted nucleic acid is what an infectious disease is, right? If you have an infection from HIV, the disease comes from the unwanted disease, HIV nucleic acids. Same thing with anything. So very often we would like to set up something called a beacon, which I'll describe briefly. It has a fluorescent molecule, but it's got this little hairpin structure. And because it has the hairpin structure, the fluorescent compound is next to a quencher. There it is sort of in more double helix form. So being the fluor next to the quencher, the fluorescence is not displayed because any time the fluorescence tries to get started, it's quenched by the quencher. But if you have DNA here and you make a loop, say, with T, C, C, G, A, T, knowing that T pairs with A, and C pairs with G, and C pairs with G, and G pairs with C, and A pairs with T, and T pairs with A, the unwanted DNA, say from the virus that's infecting you, will bind to this. It will break the beacon open, and it will cause the fluor to be a long way away from the quencher, and now the thing will glow if you have green DNA there. Well, okay, so the thing wrong with this is, of course, if you've got a lot of DNA in your body and the stem is holding the beacon together is made out of G, C, T, and A, there's bound to be some DNA somewhere floating around in your blood that's complementary to the stem region. And so the DNA will be invading that stem and separating the fluor and the quencher, causing the fluor to glow. And that means that you will get what we call in the business a false positive, which, if you can imagine, the only thing more annoying than being told that you have an HIV infection is to be told you have an HIV infection when you do not, right? And so this gives you background and false positive. And so by putting funny bases, and we are referring to these as components of an artificially expanded genetic information system, AEGIS, that's where that acronym comes from. Of course, the minute you have the stem held together by PZ pairs or SB pairs or whatever, you don't have to worry about that beacon being invaded. And in fact, that's a remarkably useful tool. But this is actually a branched DNA nanostructure that is actually used to measure viral load in patients' bodies. People who have HIV, hepatitis B, hepatitis C will actually use this assay where people are trying to detect, say, 8 molecules of the virus per milliliter in blood, and we actually put the non-standard artificial nucleobases here and here to improve the signaling of the nanostructure suppressing noise just by making this assembly, which is many signaling units per one capture of one target DNA, not be invaded by the natural DNA. This is a $100 million a year product, and I don't want to encourage any of you to get HIV or hepatitis, but if you do, you might have the pleasure of knowing that a small fraction of what your healthcare provider pays to personalize your care comes back to the Westheimer Institute and the Foundation for Applied Molecular Evolution to support our research. So we're actually quite happy about this. But again, So that's how the artificially expanded genetic information system pairs. We get crystal structures, it finds orthogonally, it does all the static things that you expect from nucleic acids. But remember, life is a self-sustaining chemical system capable of Darwinian evolution. So one of the questions is whether a 6, 8, 10, or 12-letter DNA, remember, you just have a 4-letter DNA, so you're inferior. 6, 8, 10, or 12-letter DNA, can it evolve? And particularly, can it adapt? And while we're at it, we might as well get it to evolve and adapt to do something useful, like bind to cancer cells. And so here is a relatively simple scheme. It was developed initially by people like Jack Szostak and Jerry Joyce and Larry Gold and Andy Ellington, where you start by taking a library of many nucleotides containing G, A, C, T, Z, and P. We're just going to do the 6-letter version tonight. The 8-letter version is too complicated for the evening. And, you know, that's a random variation. We'll bring along some target cells, say cancer cells, and we'll mix the library with the cancer cells. Now, of course, most of the library members have no function, so most of them do not bind to the cancer cells, but a few of them do. And so you can wash away the ones that don't. And when you do, you recover a few that do. You separate them. These are the so-called survivors. Now, these are DNA molecules built from a 6-letter genetic alphabet, G, A, C, T, Z, P. The Z has a nitro group on it for the chemist. There's extra functionality. We've got more stuff on the extra letters. So that means they have more functionality. They're more likely to have some in that library that bind. And now the evolution has fun, right? We made GACTZP, 6 nucleotide. We then let them have children. Remember, DNA can be copied, the copies can be copied, the copies of the copies can be copied, and the copies of the copies of the copies. I'll stop there. You can make a lot of the copies of the survivors with mutations, sometimes, where the mutations are themselves replicatable. And now what you do is you get a new library, but this new library is no longer a random library. It's one that has evolved under the selection pressure that we must bind to the cancer cell or else we'll get washed away. So this is an in vitro laboratory evolution experiment. So the new library is the descendants, the kids, the children, if you will, of the survivors who've survived the selected challenge, not avoided being gored by the mastodon, but rather the ones that have been able to stick to the cancer cell. Okay, so now what you can do is repeat the cycle, this time, of course, with a library that is enriched in molecules that bind to cancer cells. And, of course, in the next cycle, what will happen is the fit children, the ones more likely to survive the selection, the ones that are more likely to bind to cancer cells, will survive even better. If you do a number of these cycles round and round and round, I won't even go there, after a while, you'd begin to see all of a sudden up come some cells, some molecules with a 6-letter DNA that bind to cancer cells. This is after 11 rounds of selection. By the way, this is quite fast with just standard cells, standard DNA, 4 letters, not a lot of stuff, right? Certainly not a nitro group and not a lot of stuff that we have on Z and P. It takes you 18 or 20 rounds of selection to even begin to see this kind of. So the 6-letter DNA is evolving faster than 4-letter DNA. And eventually you can go back and get some of these. We can determine their sequence. There you go. There's the sequence of a DNA molecule which contains Z and P, which I put in red, which binds to cancer cells. This is a useful diagnostics. More important, we make it. This cell, to explain what this plot is, this is the intensity of a cell, and this is the number of cells having intensity. What do I mean by intensity? Well, we put a little fluorescent marker on the molecule that has evolved out of the selection pressure. And so if the cell binds a lot of them, the cell has a very strong green glow. Actually, if we now start removing the Z's and removing the P's, that is removing the funny bases, going back to just the 4 letters, tend to lose the binding to cancer cells. That is, if remove the Z and P, the binding goes away, then Z and P is important for the binding. By the way, we can do more with this. I mean, we can actually take extra letters in the nucleotide alphabet and write extra proteins and amino acids into the protein lexicon. This is actually work that was done by Chris Switzer and Jim Bain, now close to, good grief, I'm getting old, 20 years ago. The horizon is really unlimited with these extra cells. Now remember, reproduction is alone not sufficient to support Darwinian evolution. It's got to be reproduction with mistakes, reproduction with errors, where the errors themselves are reproducible. That's the key element here. So yes, in general, A pairs with T, G pairs with C, Z pairs with P. Your biochemistry course is going to be a lot more complicated. But never mind, S pairs with P, V with J, K with X, most of the time, but sometimes mistakes are made in this copying. Those are the mutations from which you can actually do evolution or selection within this system. And that's, of course, what makes this Darwinian. Well, for those of you who are science fiction fans, if you listen very carefully to E.T., you'll discover as E.T. is dying for the 8th or 9th time in that film and being brought back to life, the kids crying each time, you can hear them saying that E.T. has a 6-letter DNA alphabet, so this actually has precedence in the science fiction literature, not as old as Mary Shelley. I should say that you then listen to the chatter, they say one is diaminopurine, not diaminopurine, and the other is a pyrimidine which we cannot identify, which means that these were not very good scientists. This picture, by the way, cost me $500. I had to pay Steven Spielberg to let me put it in the book. So what about risk? Okay. What about risk? Okay, so you say, hey, we've got a 6-letter genetic system that is not quite self-sustaining in terms of evolution, but is capable of evolving. And I love to put this risk diagram up because this is actually Eric Schmidt, who is the biosafety coordinator of the European Union, who made it to the following point, that if you're dealing with an organism which has a alien genetics or an alien core molecular biology, it actually is not quite necessarily toxic. And he draws this Venn diagram, okay? So within the red circle are all organisms that you might construct that have standard Terran biochemistry. Outside the red circle, you do not have standard Terran biochemistry. So these are interesting parasites. Remember, you and the most nutritious food is the food that's most like you, right? That's just because you are looking for nutrients, and so you have to get the nutrients from. So meat is more nutritious to you by this logic than grass, for example. But the argument now is that if you are using standard chemistry, right, the organism that uses it is a parasite because he finds you tasty, right? So we are, of course, in our system entirely outside of that. We're making something unnatural, which is why Eric Schmidt says this is a xenobiotic and why it's a, quote, ultimate biosafety tool. Okay, now, within the green system, of course, is capable of evolving. Outside the green system is not capable of evolving. Of course, if the thing is not capable of evolving, it really is hardly a risk at all, right? Because, if it can't evolve in response to challenges, it will eventually die. Inside the circle here is self-sustaining. Outside the surface, it's not the blue circle now is not self-sustaining. That means it needs to be fed. What we're trying to do is, of course, going back through the system where we're trying to use unnatural stuff. We're trying to make it capable of evolving. Of course, we're a long way from getting it to be self-sustaining. Craig Venter, however, as you may know, with Ham Smith, produced a cell which uses standard Terran biochemistry. It is self-sustaining and it's capable of evolving, so it's right squarely in the middle of the risk circle. You just have to remember what Craig synthesized was really not materially different from what is already out there in natural biology. So it may be risky, but it's not a new risk. So there you go. So I've told you now about observation, and of course exploration is part of that. We've talked about analysis. I have not talked about origins. We've talked about synthetic biology. What we can say is that, yes, we can construct, okay, using a scientific method that is more complicated than what we were taught in school, where our biases are jarred by exploration, for sure, no question about that. Natural history certainly is expanding our view of what life could be like, that is RNA life only before proteins emerge. That's a possibility. And science, of course, is this human intellectual activity which has a process that is, in the field, appropriate way, able to dislodge the scientists from always coming to the conclusion they set out to conclude. And synthesis in this respect is the ultimate tool. It drags this scientist across territory, uncharted territory, where they're forced to solve unscripted problems, and if the theory is inadequate, it crashes. And therefore, synthesis can drive discovery and paradigm change as well, better than analysis and observation by themselves. So what is life? Well, still don't know, but we seem to be able to get something that we value in life, adaptation to perform a function, here binding to cancer cells is the example that I showed you, from a functionalized nucleic acid with extra letters in the nucleic acid alphabet, designed by synthesis, made by chemists, able to replicate with imperfections, of course, where the imperfections are themselves replicable and therefore able to evolve. Now, we talk about life being a self-sustaining chemical system capable of Darwinian evolution. You say, but this is not self-sustaining, right? But of course, that's why we emphasize system in this case. And of course, the differences between this and a 12-letter Darwinian DNA system capable of assisted DNA, assisted Darwinian evolution in a test tube are more than obvious. So with that, let me stop. I'll thank you for your attention, and I'll be happy to answer any questions you might have.
Well, thank you, Steve, for that very thought-provoking lecture. We have time for questions. Would you like to moderate your own from up there?
I'd be delighted.
The one thing that you should do, though, is to repeat the question when asked in case people can't hear.
All right.
To save me running around with a microphone.
We got a hand right there. Yes, not try to, yes. Say loudly and I will repeat the question.
Okay, so in your lecture, you're basically expecting a carbon-based life throughout your lecture. But there are some hypotheses that silica-based life can also one of the carbon, chemically carbon.
Yes, where's your periodic table in this room? They don't have one. Absolutely. Well, John Barris and I wrote a book under the auspices of the National Academy of... Oh, yeah. So the question is being asked is whether or not... I'm making the assumption that carbon-based life is what we're doing here. You saw a lot of carbons out there, and you saw no silicons, right?
Your definition of life, based on those machinery used earthian life, like the protein, RNA, or possibility.
Well, that's right. So the definition of life as a self-sustaining chemical system capable of Darwinian evolution is a theory of life, right? It makes the assumption that the only way that matter can be organized in a way that gives us properties that we value in life is through Darwinian evolution, this process. But it does not make the assumption that you don't have silicon, right? If you can, tellurium instead of sulfur. There's all sorts of analogies that you can make. Arsenic instead of phosphorus is one that we worried about a lot. So there's no question that if you can construct a working system which gives you Darwinian evolution out of silicon, that will pass the muster for life by this model. It's a theory of life. It's a statement. It's deeper than a definition. It's basically a statement saying that this is the only way we think it's possible to get, Now, keep in mind that if you ever, and you presumably as a human species are not far from this point, if you ever get to the point where you're able to do germline manipulation so that you anticipate future things and change the DNA and the germlines that are producing your babies, this is something that is not allowed by Darwinian evolution. You're not allowed to have prospective mutation. This would be called maybe Lamarckian evolution. Of course, the question is when the human civilization adopts it or creates that technology, will it cease to be alive, right? That's sort of the paradox of this. But yes. But the answer is silica. No, we've tried. There was John Barris and I, we put together a report with the National Academy on Weird Life, as it was called, and it was published in about 2010. You can get a copy from the National Academy, where we tried to come up with systems based on silicon, based on avoiding as much carbon as possible. And one can conceive of these. They have never actually been synthesized. But are you a synthetic chemist?
I was a biologist.
Oh, okay. So he's going the wrong direction, right? Yeah, another question. No other question. Oh, in the back. Oh, well, yes. Yeah. Yeah, absolutely. If you go out and find data. So the question is whether a robotic system, right? If you go out and find data in the cosmos, would you say that data is life? And that's, of course, the subject of many Star Trek Next Generation plots. And the answer is, we would probably say he's a biosignature, as he was the evidence that something that was life made him. But that's only because we can't conceive in our feeble way of looking at it of him coming out of spontaneous generation from a soup. But that's just us. But yes, anything that's capable of Darwinian evolution, even if it's a robotic structure, would be called life at this. In the back. Yes. An unscripted hypothesis. Sure. Well, I mean, keep in mind that what we do as science when we're doing routine science is we have control over the hypothesis that we choose, and of course anybody, especially if you're trying to write a grant application, does not pick a hypothesis that is difficult to test. So you will tend to have scripted hypotheses. And the science fairs are well known for these, right? You know, is my cat left-handed or left-pawed or right-pawed? This is a common thing. But with synthesis, right, you're being driven by the goal. And so you don't have the option to pick and choose your hypotheses. The hypotheses that you must deal with, the theoretical elements that you must deal with, are the ones that you're relying on to get that synthesis to work. And so that's what we mean by unscripted in that context. Yes? Yes. Mm-hmm.
Why is there not a big piece of free prong, piece with holes? Free prong and thick holes?
Yeah.
Why can't we just go up and
Absolutely. No, if you would like to do that and have there's a, no, the problem fundamentally is that with carbon, nitrogen, oxygen, and hydrogen as the only elements from which you can build, it is actually very hard to build a small thing with 3 prongs or 3 holes without having a charge on that particular molecule. The big things actually you can do. So the reason why you only see 12 bases and 6 base pairs instead of 16 bases and 8 base pairs, 2, 4, 8, 16, which is what you would expect, it has to do with the constraints of chemistry associated with how many bonds carbons, oxygens, nitrogens, and hydrogens make. So it's very core to the chemistry. So you cannot get this from the prong story. You have to go back and look at those structures. So that's a very good question, however. Very observant. I have to worry about this person. Yes? So let me summarize that for those in the back. Absolutely. So the question is, right, Miller and Urey made up a model for what they thought the early Earth atmosphere was like. And so they put a lot of energy through it and got a lot of goo out, tar, and then eventually they could get out of that goo some amino acids. And that's a very important initiation of pretty much modern prebiotic chemistry. So the question is, well, we can go to other planets as well, and we might be able to make models about what their environments were, do the same kind of experiment and get different. I mean, we can go to a planet which is dominated by silicon hydride in the atmosphere and try to get silicon life out of this, which would be, and your problem is that, yes, I mean, I would love to do it. Keep in mind that almost all of the problem has to do with the uncertainty of the ancestral atmosphere and the ancestral environment. So ASU has, of course, a wonderful astrobiology organization which is very much involved in this, and you know, they will tell you, oh, I mean, I don't know, Jack, I mean, some days it's Mondays, Wednesdays, and Fridays, it's well, the oxidation state is more and less of it. You know, it depends on the model. So that's a large amount of uncertainty, meaning relatively little constraints on what you can actually get. Now, Titan, for example, is a moon of Saturn. It's got a lot of organic stuff going around it, and you know, I mean, it's a rich source of organic molecules, and I'd love to get a life form that could live in methane liquid at 95 Kelvin. That's about minus 200. Yeah, exactly. So your Earth, unfortunately, has first been poisoned by these plants. They put all this oxygen in the atmosphere, and that it really destroys the ability to do good organic chemistry, right? Because anything that you put out onto the table gets toasted, right? The butter goes rancid, the bread becomes toast eventually. And so you have a problem with this. And then, of course, you know, we have a big fight going right now as to what minerals were available on early Earth. I mean, I'd love to have borate minerals. I'd love to have gypsum. I would love to have molybdate minerals. We'd love to have these in a dry desert environment, not actually unlike Death Valley, and not, so that matter, not unlike Phoenix. But then I got the geologists telling me that their models for early Earth have so much water on them that there was no dry land, and so I can't have a phoenix. Everything's submerged as a Kevin Costner water world. And so you'll find that when you try to model what the early Earth looked like and then what early Venus looked like or what early Mars looked like, you'll get consensus on the direction. Almost everybody will agree that Mars had more water then than it has now. Venus was more habitable then than it is now, but you won't get a lot more constraints in that until we do a lot more work. Yes? Yes. Yes. And we're trying to get them both in the same, we're trying to get the functional stuff and the information in the same molecule. Yeah, maybe I missed it presenting it, yes. Yeah. Yeah. So the question is, I mean, why do we take this chicken and egg problem seriously? So right now you have, of course, a 3 biopolymer system. You have DNA that passes information from you to your kids. You've got proteins, which does most of the stuff that needs to be done. And you have RNA, which carries information from the protein, from the DNA molecule to the ribosome to make the proteins. And your problem now is the creationist irreducible complexity problem, right? No one knows how DNA arose from prebiotic soup. No one knows how RNA arose from prebiotic soup. We even having problems getting proteins out of prebiotic soup, although these tend to be a little bit easier. How you would get all 3 of them at the same time, where one is performing the genetic role, one's performing the functional role, and one's the messenger between the two, is just a problem that is a complexity, irreducible origins problem. So everybody was very, very happy when Tom Cech and Sid Altman back in the 1980s discovered that RNA, in addition to being a genetic molecule, and there are RNA viruses, for example, could also do catalysis. Because we said, hey, the problem is solved, right? We don't have to get proteins and DNA and RNA and ribosomes and machinery to all rise immediately to get life going. All we need is an RNA molecule that will itself be a catalyst and specifically catalyze the template-directed synthesis of another RNA molecule, its son or daughter. And then life would get started. Because that RNA enzyme that catalyzes the synthesis of RNA from an RNA template would be imperfect, but the imperfections would be replicable, okay? And so that's where this story goes. That's why we look for a single biopolymer. So it could be proteins. People have argued that proteins were the primordial biopolymer, RNA came later. The only problem is that proteins don't have any easy Watson-Crick, big, small, prong, hole replication potential. And so, that's why RNA is better. At least with RNA, I can imagine replication principles. So that's where, is that clear? So we are up against a chicken or egg problem where we have this very complex system, which is your modern metabolism. We had to simplify it. We had to simplify it in a way to get one molecule, do both catalysis and genetics, and do stuff as well as encode stuff. And we get one molecule, then we have to worry about that one molecule arose. And that's a hard enough problem without having 2 or 3. Yes. Yes. So we're using the property of a 12-letter, or in this case, 6-letter genetic system, putting it under evolutionary pressure where for part of it, it's folding to bind to cancer cells. It's doing stuff like a protein, and then another part of that cycle, it is being copied like a genetic molecule.
2 more questions, one of which is...
No. Yes, sir, shoot. Yeah, I guess so. Yes. Yeah, I mean, keep in mind that there's 2 questions you're asking. The question is whether they're more error-prone intrinsically or whether they're more error-prone in our hands. Yeah, so intrinsically, actually, they are probably less error-prone because the AT pair joined by only 2 hydrogen bonds is weak, and all of the funny ones that we have are joined by 3 hydrogen bonds. So this is a very, very, very error-free system when it's just coming to hybridization. The problem is that when we make copies of it, we actually use enzymes, which we get from natural biology, These are enzymes that have evolved for billions and billions of years to take GACT and not ZP, SP, or any of these other things. And so those enzymes, we mutate, we evolve them, we do engineering on them, we do all sorts of stuff to try to get them to handle our funny alphabet. And so those are more likely to make mistakes with our funny stuff because they're unnatural, not because they're intrinsically bad. Yes, Paul?
You raised the question about whether making new forms of life is risky, and you've persuaded us that the risks are very low. And then the flip side is it useful? And you've given us some examples of what's been done. But this is presumably the tip of a huge iceberg, and the future of synthetic biology must open up all sorts of wonderful possibilities, possibly further risks. Can you just, in a few sentences, say, what's next on the agenda for
Sure. Well,
novel
keep in
forms
mind
of
that
life?
synthetic biology, and I mean, it's somewhat embarrassing because as a good chemist, organic chemist, synthesis is what we do, is not a field. So when I woke in 2004 with an article in Nature and in Scientific American telling me that Pete Schultz and I had both invented synthetic biology together, I called Pete and said, "Did you invent synthetic biology without telling me?" And he said, "No, but we're going to write it into the next grant application." But synthesis, of course, is an old field in chemistry. Almost all the compounds that you have in your clothing as dyes are synthetic. And of course, biology became synthetic in 1970s with recombinant DNA technology. And there was a big concern at the time as to whether or not this would lead to plants that would escape the laboratory, bugs that would escape the laboratory and be harmful. There was a huge conference in Asilomar in California where rules were set out. And over the last 40 years, we've come to pretty much understand that there's not a lot of risk associated with this, but there are still some people who don't like genetically modified organisms. So right now they'll make plants, right, which will be resistant to herbicides. So you plant the plant with herbicides and you put herbicide down and all the weeds die and the plants grow better and they're cheaper. So, and I mean, a good biochemist will tell you, and I will be one of them, that this is not materially different at all from the plant that you get in it. But the future is hard to say. I mean, the future, basically, we are desperately trying now to get E. coli to metabolize steps here that will enable us to get the Z and P, for example, made inside of an E. coli cell as triphosphate.
Right, I was going to ask you, can you upload this stuff into cells and let them take it away?
Yeah, Stefan, who's your guest in the chemistry department, actually has contributed one of the enzymes to one of the steps in that metabolism. So we have that metabolism working in E. coli. I did not talk about it because it's a little premature to speak of. But yes, to get an E. coli that's able to maintain plasmids that contain funny bases. And of course, the funny bases now will be used in diagnostic systems. And so you'll have an E. coli package that is able to do a complete diagnostic workup of you, right? And so you don't have to pay money for each individual diagnostic. That certainly is in the...
So unlike dogs, which are human creations after all, yes, but very close to wolves, we're dealing here with truly novel organisms that could, in the very near future, be autonomous. And
yeah, that's a big problem. The autonomy of these things, especially, is also true with just recombinant DNA organisms, is less than we would like. They are dying, even when we try to keep them alive. But in principle, yes, you could have autonomy.
Right.
So the problem now would be that this poor guy, you drop him out in the world, he wants to go find some Z to eat.
That's right.
And he won't find any.
Not yet.
Yes.
Not till we've created a whole ecosystem.
Yes. Life finds a way.
That's the next step. All right. Well, I think we're going to have to draw it to a conclusion, not least because you need to sign books outside. So before we wrap, these occasions take a lot of organization, and my staff, particularly Adriana Fontes, who ought to be around somewhere, and Katie and Alyssa at the back there, you'll have met them on your way in, and several others who have helped out over the weeks in promoting and dealing with the website and encouraging you all to come. And so I think a round of applause for them as well as for our speaker. And see you all next time. Drive safely.